We're talking with Professor Arpad Pusztai. He's in Hungary, and he has been doing lots of research on plant lectins since 1968 at the Rawat Research Institute in Hungary, and he also began doing some work a few years back on genetically modified organisms in potatoes and rats that we're going to talk about. Where are you located?
Well, at the moment, I am located in what we call in our summer home. We refuse to get closer to the universities where we teach. My wife teaches about 400 kilometers from this spot. My wife is a professor of nutrition, and I teach at a doctoral school. Mainly molecular biology and genetic modification.
Genetic modification, yes.
Yes, I try to point out all the things that ought to have been done before these things had been commercialized, but they hadn't been done. And it is a totally scientific approach. We know what ought to have been done, and I just point these out. Incidentally, Hungary has a moratorium on growing Monsanto 810 corn. It's not allowed to be grown in this country. And Hungary is the second largest corn producer in the European Union. And this is what I said, that I'm teaching doctoral students, PhD students, to point out all the things that have not been done, although they ought to have been done, risk analysis, safety studies, that ought to have been done before commercialization.
Before genetically modifying these foods, yes, sir.
Yeah.
And you started your research many years ago, I think, 1968, but it was mainly on this protein that we know of in this country called lectins.
Yes.
Correct?
Yes, that's it. That was my background.
And what are lectins exactly?
Well, these are sugar-binding proteins. They are sort of communication proteins. So anything that needs to tell something to that cell and coming from the outside, it will be communicating via the sugar residues, and the lectins are the natural counterparts of that.
What role does lectins play?
They do sort of impart messages to the immune cells, other cells as well, but it is all the lymphocytes and other things, like, these communication molecules in the body, which tell the immune system to do something. They're all lectins, a fundamental building block of life. They're always talking about that the various Bt toxins, there are several of them, they seem to be claiming a selectivity for a particular insect. This selectivity, this claimed selectivity, is a fictional business.
I see. So as much as they would say, what you're suggesting, that this 810 selective lectin just for this particular corn borer actually affects butterflies and other species.
Or some lectins, based on selection, can actually be insecticidal. I give you the best example, all the Bt toxins. This is the Bacillus thuringiensis toxins, which are by Monsanto and others put into corn and all sorts of other things like cotton, and it is they transferred the gene of the Bacillus thuringiensis, one of the Bacillus thuringiensis toxins, and that reacts with the insect gut. The reaction is very harmful for the insect.
I see. So what Monsanto, let's see if we understand. Monsanto says this Bt toxin 810 is specific to this European corn borer, for example, and it won't hurt anything else or react with anything else. This is the way we've created it. And you're suggesting that your research shows that's not the case.
I think that's a very restrictive view of it because there is no absolute selectivity. It's the insertion, in fact, causes mutation, and this is what is called insertional mutation.
I see.
You are shooting blindfold, and by selection, you're trying to pick out the ones which will be suitable for your purpose. But you cannot say where it landed, and you don't know what sort of reshuffling. You see, this is like a pack of cards which is reshuffled, and you don't know what is going to happen. And normally, it is now reasonably well known that something like between 2 to 5% of the plant's own genes are affected. We don't know which way they are affected. We have found methods which will tell you this, but you cannot say it in advance that I am going to shoot this thing into a part of the genetic network where there is not going to be any problem.
And they genetically modify this into the corn or the cotton or something.
Yes, that's it.
Uh-huh.
And this is probably one of the best known examples, but they never talk about it that these are actually lectins.
Toxins that they put into the corn with the idea that they will kill the insects that eat the corn are actually lectins.
Yes. So we could find a lectin, an insecticidal lectin, which would do away with that damage by aphids by simply poisoning the aphid, because the particular lectin that we have transferred by its gene into the potatoes, when it was expressed in the potatoes, did in fact harm the aphid. But this we can do quite well. What we could not do was to do this very selectively. So when we found, for example, the aphids are controlled by what you call ladybugs.
Mm-hmm.
And when we found that when the ladybugs would eat the aphid,
yes,
was just as much poisoned.
And we need the ladybugs for the whole ecology.
Yes.
Yeah.